And it's a pleasure to have Nikki Sanderson from Brown speaking this afternoon about structure and neural correlations during spontaneous activity.
Awesome. I'd like to thank the organizers for inviting me. This is my first time at the Fields Institute and in Toronto, so real honor to be here, so thank you. I'm excited to share about this true data analysis research project, a collaboration between me, whose background is in topological data analysis, and experimentalists who really work in the lab. And I just want to mention, first off, this is a collaboration with Enrique Hansen, who was a postdoc in Herman Sumbre's lab. Sarah Noren is the engineer that did a lot of the immunostaining, and Virginie Condant is the lab facility manager. So with all of this data, it takes a total village to even produce data, let alone analyze it successfully. So a big shout out to my collaborators, because I came into a project that had 5 or 6 years of work to just produce data that I was then able to analyze. So again, also very honored and privileged to work on that data set. So just bringing a little bit of the lab to us theoreticians. This is a bit of what the data that I get looks at. And what we're looking at right now is this part of the zebrafish brain called the optic tectum, and that's part of the visual system. And what we're seeing here in these different colors are different types of individual neurons. So it's really flashy, it's really cool data. And one reason we're interested in this optic tectum region of the zebrafish larva is because it's known that there are these groups of neurons that we call assemblies that are localized in a region of the tectum, kind of like those shown here. And they respond to very targeted visual input. If you have light coming into, let's say, the right eye, there's a little group of neurons on the left side that's going to all kind of co-activate together. And if this light changes and is now more in the front of the head, a different group of neurons that are also localized more towards the front of the brain, the optic tectum, is going to kind of co-activate together. So this is something that we've seen before, this kind of tiling of the optic tectum with these groups of neural assemblies. So here is just a picture of the calcium imaging. We see that the neurons are lit up. One of the reasons we're studying this Donio rario or zebrafish larva is because when they're babies, they're actually see-through. So they've been genetically modified so that their neurons emit light when they fire. And so these are actual pictures of the inside of the brain showing the light when this fires, when the neurons fire. And what's really amazing is that even without this external input of a stimulus, they've observed these groups of neurons spontaneously activating in the brain without any stimulus. So these are just something internal to the wiring is leading to this co-activation of these groups of neurons. And this is the time where I get to show you this video. And the thing that I did not do in my preparation for this talk was pull out the video to show you. So you're gonna just have to trust me on this one, so you don't need to see my desktop, that this group of neurons here, we're gonna start with a black screen, and they're all gonna kind of just come on together and light up and then go off together. And there's a little bit of randomness in it. It's not exactly everybody goes on at the same time and everybody shuts off. There's a little bit of twinkling that's happening. And again, this is happening without any external input. And it's this spontaneous activation of these neurons that I'm happy to show you the videos of later, because it is very cool, that we're interested in studying. And so the data that we have is a recording of 12 different fish for an hour in a dark room with no visual stimulus. We're getting calcium imaging, so we're getting the change in the fluorescence of each individual neuron across an hour in time. That's this trace up here. We then do some pre-processing to smooth out this signal. And what's happened is we've kind of identified points where we believe that these neurons are spiking. And then this kind of decaying exponential is kind of representing the slow decay rate of the calcium in the neuron after firing. So these are what the signals we're looking at. And we have one time series per neuron. And we have a recording over an hour. We record at 15 hertz. So we have 54,000 time points. And we get about 2,000 neurons per fish. So all in all, per fish, per recording we have 2,000 neurons by 50,000 time points data set. where the content of that matrix is going to be the smooth calcium signal, indicating neural activity. And then the other really cool thing that we have is an identification of these different neuron types from immunostaining. So some neurons are cholinergic, glutamatergic, or GABAergic. And we know that. We know what are the different neurotransmitters that these chemicals release and how they then affect the neural circuitry because of, or the neural circuit function from that. Okay, so the first thing we need to do from this 2000 by 500 or 50,000 matrix is identify which neurons are actually part of these compact assemblies. And this isn't something we're going to necessarily like want to do by eye. So we just use a kind of out of the box method called PCA Promax. And this is going to identify neurons that are functionally similar, but then we want to go a step further and identify those that are also just like local in the tectum. So here we find a group of neurons that are functionally similar, but they're spread across the tectum. We want to find the ones that are compact. And so we look at just the entropy of the distribution of pairwise distances, and that's just going to give us some type of thresholding to say, oh, this is a compact assembly. So now we've identified a bunch of different compact assemblies. And our goal is to understand the spontaneous dynamics of these compact assemblies in the tectum. The way we're going to do that is by looking at the pairwise correlations of the neural activity for each of these compact assemblies. And some questions we have are, how are these correlations between these assembly neurons different from just random neurons we would pick in the brain? What about other controls? Is there some sense that this activity is low rank? So this is what we're going to be look. Oh yeah. Yeah. So actually, I didn't tell you the first time. So let me tell you now. So we have these time series per neuron. The way that we're going to get this neural correlation matrix is just by taking a dot product between the two time series. Thank you. That's a really important question. And the reason that we're just taking a dot product and we're not doing a Pearson correlation is because if you look at our signal, it's a non-negative signal, right? We're talking about positive calcium activity or no calcium activity. And if we were to do something like Pearson correlation, we would be mean shifting. And then we would have high correlation between neurons that are co-silent, whereas we want to focus on, because this is such a sparse signal in terms of this hour-long recording, that's not like they're spiking all the time. We want to identify in our correlations neurons that are coactive together. And so that's why we're using just this dot product of this non-negative signal versus something like Pearson correlation. Yeah, and it's a really important thing, depending on your data and the questions you're asking, how do you go from time series to correlations is very data set specific. And that's what we're doing here is that dot product. So in particular, these correlations are non-negative. Okay, so we're going to look at a few different matrices coming from this data. We are going to look at the total neural population. We're also going to look at the correlations of these compact assemblies. We're also going to look at controls looking at random collections of neurons and random matrices with distributions that match those of the correlations from the actual data. We'll talk more about those shuffles in a bit. We're also going to be looking at the correlations when we restrict to the time periods when the assembly was active and then when the assembly was inactive. So we have an algorithm that goes through and detects when is an assembly on, when is it active. We need so many of the neurons to be firing, and we need that firing of those assembly neurons to be more dominant than other population activity, because if those neurons were firing along with other population activity, it might not be an activation of that particular assembly. So we have criteria to say when an assembly is active, such as like right here, this is an on time period. And then this is off. So we're going to separate those two times and do correlations separately, see if there's a difference there in the structure. And then finally, what we're going to do is we're going to do a sliding window analysis through this onset of activations that we've detected and see if we could tease out any information from the correlations via that sliding window analysis. That's what we'll talk about today. Okay. So if we just look at the entire like 2000 by 2000 neural correlation matrix, we could ask if we see any structure jumping out at us just from like observation, and it's really hard to see. So one thing we might do is look at the distribution of pairwise correlations between different cell types, look at their correlations during activation, and compare it to the random controls. And we don't really see any strong trends jumping out. Nothing really looks distinct here. And so another thing we might consider doing with pairwise correlations is to look at something like the singular values. And one thing that the singular values can really detect is if we have an assembly correlation matrix and we keep the values, but we shuffle where they are in the matrix, this is going to preserve the distribution of correlations, but it is going to change the singular values. Yeah, because we're really changing the internal structure of the matrix. But something that the singular values are also very sensitive to is monotonic transformations of the data. So if we have the original assembly correlations and we just make the stronger ones stronger, but we have a monotonic transformation, this is going to change both the distribution and the singular values. But in some sense, the structure of this correlation matrix, even with this monotonic transformation, it's still in some sense preserved. And this is something that is important to us. And one reason that it's important to us is we're using this calcium signal that's been super smooth and pre-processed as a proxy for neural activity. So we're kind of assuming that there's this underlying monotonic transformation happening, which is why we don't want to use traditional methods like singular values. So instead, we're going to propose this click topology pipeline that's going to take a correlation matrix and it's going to output Betti curves. And we're going to use the Betti curves to look at structure in the correlations. So some things that click topology is invariant to is reordering the neurons. And singular values is too, right? We know if we kind of shuffle the rows and the columns to try to see if we could identify structure in a matrix, we're going to get the same singular values. We're also going to get the same Betti curves out. Unlike the singular values, click topology is invariant to these monotonic transformations. So if we take an original correlation matrix, we monotone transform it, we see the Betti curves are identical, but the singular values change. So sometimes singular values might be giving some indication of a structure or not giving indication of a structure. And that's really coming from some type of monotonic nonlinearity potentially. And so this is a reason that we might want to be using this click topology is because it's invariant to this type of transformation that we see all the time in neural data. On the other hand, the click topology is really sensitive to the organization of the correlations in this matrix. So when we shuffle where these correlation values are, we see that the Betti curve signature really changes. So for instance, the actual assembly had no Betti 2 shown here in red. It was just a flat line 0. And the second we shuffle it, we see we have just like a ton of Betti 2, which are just voids, and a ton of Betti 1. However, the singular values, they changed a little bit, but not as sensitive. So just to kind of restate what I've said before, click topology is in particular very sensitive to the reorganization, whereas the singular values are less sensitive to the organization, but very sensitive to these monotonic transformations, which we really believe are underlying our data. Another reason that we like these Betti curves as a summary of the click topology is that they have a very stereotypical signature when we have random symmetric matrices as our input. And what we see, and I think Nico talked about this earlier, sorry for the change in colors, is that the Betti curves, as we increase the homological dimension from Betti 1 to Betti 2 to Betti 3, the maximums of these Betti curves increases in value. Yeah, and this is really different than if we look at geometric Betti curves where we're taking points from some Euclidean space and looking at their pairwise distances, using that as the input to the click topology pipeline. There we see the opposite trend in the maximum values of the different dimension Betti curves. And so this is one way that we could tease out truly random from random geometric structure. Another kind of type of null model that we might want to consider and see like, oh, does our data show us Betti curves that represent something low rank, would be to look at the correlation of points in low dimensional spaces. So here we're thinking about points as vectors and just taking their dot products. And if we take points in any and all orthents of space, we get Betti curves that have very little Betti 1 and Betti 2, but it's non-trivial. But it's definitely less than the random and possibly the random geometric. But if we only take points in the positive orthant, which is where our data lives, right? Because we're taking dot products between 54,000 dimensional vectors that are all non-negative because they're the positive calcium traces. We're talking about data that looks like this. And here, we don't see any Betti 1 or Betti 2 emerging. So it's not just often a matter of like, oh, you're looking at a low rank model or you're looking at a random geometric model. There's nuance to that too, right? When we talk about random geometric, we're talking about, in this case, Euclidean space that we're sampling points from. It would be a different null model if we sampled from hyperbolic space. And this is just kind of to like highlight, okay, if we were to do this kind of low rank thing by taking dot products of vectors in low dimensional spaces, we still need to be aware that there are choices to be made. And it could change the topological signatures that we see. Cool. I do want to point out here that in all cases, it's not really obvious here, in both the mixed and the positive low rank models, for rank 1, it's theoretically proven that there is never any higher dimensional homology beyond 0. So you will never have holes or voids. So that's a unique signature of rank 1. Is that like across the mixed and the positive? Yeah, cool. Okay, so hopefully I've convinced you why using this TDA pipeline might be interesting and worthwhile. Now let me kind of tell you what this pipeline is. So we start with our correlation matrices, and we're going to build a filtration of simplicial complexes by adding edges one step at a time, one edge at a time, in decreasing order of correlation. So the highest correlated neurons are going to be connected by an edge first, and so on. And what makes this a click topology pipeline is whenever clicks appear in this graph, where we're adding one edge at a time, we're going to fill that in with whatever simplex fills in that click. So if we have 3 neurons with all of the edges between them, we're going to fill it in with a triangle. If we have 4 neurons with all 6 edges between them, we're going to fill it in with a solid tetrahedra. And so that's going to build us this filtration of simplicial complexes. And from that, we're going to compute the homology at each step. And from there, we get the Betti curves. And we're going to, in particular, be looking at Betti 0, Betti 1, and Betti 2. And Betti 0 is counting the number of connected components. So one cool thing that I like to think about with Betti 0 is when we see plateaus in Betti 0, when it just kind of like flatlines for a little bit, it means you're adding edges between a part of the network that was already connected. And so you're saying, oh, there's a period of time where my correlations within a cluster are stronger than across clusters. So plateaus are an indication to me of some subcluster structure. When we see Betti 1, those are like these loops right here, these like one-dimensional bracelet-like tunnels that you could stick your hand through. This red blip here, this is showing some Betti 2 appearing, I think it's here. What we have here is the outside, I believe, of an octahedra, and it has a void inside, like a basketball, right? That's Betti 2, or holes of that flavor. And so we're going to be looking at these Betti curves and seeing what do they tell us about the structure of our correlation matrices, thinking in the back of our heads about these subcluster structures and these one-dimensional cycles. Okay. And to be able to just kind of summarize information from these Betti curves, we're going to go a step further and compute the area under their curves to give us an integrated Betti value. And that gives us a single number that we could then do some statistics with, which is helpful. One thing I guess I also want to note, maybe I'll note it later. No, I'll note it now. Okay, because we're working with assemblies of different sizes, this Betti 0 curve is going to start at different values, right? And so that's going to skew this kind of measure of Betti 0. So one way to kind of put it all on equal footing is we're going to use this integrated normalized Betti 0 that kind of takes into account the different assembly sizes. So you'll be seeing that in my plots. We're going to, yeah. So what does this click topology tell us about our data? Well, if we look at the Betti curves of our assemblies, which are shown in black here, and the random assemblies, which are shown in yellow, random assemblies, we just took random neurons and looked at their time series. We see that their Betti 0 decays much more slowly than if we just had shuffled the correlations, which effectively is giving us the random IID structure. So this is telling us that possibly the assemblies have more subcluster structure than just a random matrix would. The other thing we see is that there's very little on average Betti 1 and Betti 2 for the real assemblies, whereas for the random assemblies, these random collections of neurons, on average, there's much more Betti 1 and Betti 2. So that's also interesting. In some sense, that's saying that these true assemblies are in some sense acting in a more low rank way, and that there's possibly more randomness with these random assemblies. And notice that these are orders of magnitude lower than when we shuffle the correlations and get a signature of true randomness. Those were averages across all assemblies, across all fish. If we just look at an assembly by assembly basis, and we scatter the integrated Betti 0 on the x-axis and the integrated Betti 1 on the y-axis, we could see how this trend holds for each of our assemblies. And we see that, yeah, it really actually isn't just something being thrown off by an outlier skewing the average. The random assemblies really do have more Betti 1 than the real assemblies. And the other thing that we just can check is that the increase in integrated Betti 0 is actually picking up that plateau feature that we see in the Betti curve. So looking down here, we have integrated Betti values that are increasing from assembly 1 to assembly 4. And hopefully you can see this plateau becoming more and more prominent. So that's just kind of like a sanity check that these statistics that we're pulling out are capturing the features of the curves that we want it to. So now we're getting to the interesting part about like the actual spontaneous activations. So when we repeat this analysis, but now take our correlations during the activity time, we see that there is pretty much 0 Betti 1 and Betti 2 curves, which, like I said before, could be a really strong indication of rank 1 structure, not just low rank, but rank 1 in particular. However, these real assemblies, when we look at their correlations outside of activation, they look like the random assemblies, right? They have on average much more Betti 1 and Betti 2. So what that's sort of saying is that outside of these moments of time where the assembly is activated together, those neurons are kind of just acting the same as any other neurons in the brain, kind of randomly. So that's kind of interesting. So the rank 1 structure is coming from a null model where we take points randomly in the unit cube in dimension 1, which is just like a line. So you take points randomly on the line, you take their dot product, that gives you your correlations, and you look at the Betti curves of those correlations, and there will never be any Betti 1 and Betti 2. It's a theorem. So that's the rank 1 case, is that random sampling from a line and looking at the topology. Yeah, and the correlation matrix is the object that is then rank 1. Yeah. Oh yeah.
To tell you, like, yeah, you know, like 0.15. What is it? Could you just give some more intuition behind like integrated Betti numbers and how you interpret it?
Yeah, I'll go back to here. So in this case, I'm not using the actual number itself to tell me much. What I'm using it for is a comparison between two distributions to say something statistically. So actually, so the intuition between Betti 0 is that Betti 0 will be higher if the integrated Betti 0 will be higher if the Betti 0 curve decays slower. Because Betti 0 is connecting components and we're only adding edges, Betti 0 is, let me go to it. Betti 0 starts at the number of neurons you have in the assembly, and it has two options. It can either go down or plateau. And so in some sense, this integrated Betti 0 number is telling you how fast did it just go down versus plateauing. And so a higher integrated Betti 0 number to me is saying more time plateauing, more subcluster structure. Betti 1 is the area under this Betti 1 curve, and more Betti 1 means more of those cycles like the bracelets in your network. And so when we see these like Betti curves here we see there's Betti 1, so this is going to have a much higher Betti 1 value. That's this assembly up here, point number 4. Whereas this assembly 1 down here, it has this little blip of Betti 1. It's hard to see. Can you see it if I zoom in a little bit right there? So it has much less Betti 1. So it's kind of, good. Yeah, it's kind of a proxy for what's going on in the Betti curves, but the specifics of the numbers are less meaningful to me. Where it comes into play is with these distributions that we have for our different null models over here, the mixed low rank models in pink and the positive low rank models in green. We have high dimensional controls over here that are related to the data that we wanted to see. Oh, is there, we said it's low rank, but could it be actually a high dimensional structure? And what we see is that here we're showing integrated Betti 2, integrated Betti 1, integrated Betti 0 for the whole recording, during the activations on, outside of activations off, and the random assemblies. And we already see that the mixed low rank model is not fitting the data very well by the time we get to rank 3. And that makes sense because our data is not in that framework. However, even the high dimensional controls aren't really capturing what's happening with these random assemblies so well. But what we're really interested in is the true assemblies and what's happening during activations. And what we see is we have the best fit of these assemblies to the positive low rank null models. So that's kind of how we use the integrated Betti numbers to do statistics via things like Wasserstein distance between the distributions to make these claims. Okay, great. I'm going to take a sip of water and then we'll transition to one of my favorite parts of this analysis, this sliding window analysis through activation onset. Okay, so our big takeaway from that was that during activations, assembly activity looks really low rank, maybe even rank 1. Outside of activations, these assemblies look random. But what's happening at that moment of activation? To understand that better, what we're going to do is we're going to look at a half a second time window. And we're going to look at the correlations of neurons just in that half second time window. We're going to do our click topology pipeline. So we have our correlations. And then we're going to get out our Betti curves and we're going to get out our integrated Betti values. We're going to get out integrated Betti 0 and integrated Betti 1. We're going to slide our little window one time step, do the same thing. We're going to get out these two numbers. And in this way, for every activation, we're going to have two integrated Betti time series, one in Betti 0 and one in Betti 1. And when we average these over all 5,000 activations across all 12 fish, we see there's this sharp increase at the activation onset in integrated Betti 0. And at the same time, we also see this really sharp, stable decrease in Betti 1 that just crashes to 0. And what this is telling us is that something's changing in the structure of the neural correlations. This is not just an amplification of the structure that we had prior to activation. And that's coming from this fact that the way that the Betti curves are invariant to monotonic transformations. If we were to just take a correlation matrix and ramp it up with some gain amplitude, these two time series, the integrated Betti 0 and integrated Betti 1 time series, would just be flatlining because there's no change in the actual ordering of the correlations. So when we see something like this happening, that means there really is a change in the structure of how the network's relating. One thing that was kind of not as apparent was that I was showing you error bands, standard error bands rather than standard deviation. There's a lot of variability in these curves. So even though this is standard deviation, so even though we see this really strong trend of increase in Betti 0 and decrease in Betti 1, there are a lot of different ways that we could increase in Betti 0. So when we look at these integrated Betti 0 time series for 4 different activations, we see these 4 different profiles appearing. And the first thing that we went ahead and did was try to just model this and see if we could capture these different dynamic profiles with different kind of time series of synthetic neurons. So one thing we did was have all the neurons fire, same amplitude, same time. Nothing changes in the correlation. That's a flat line. This yellow line here is what happens if you just have a random IID matrix of the size of this toy network, which is 30 neurons. Model 2, we spaced out these spikes. So when the spikes are really far apart, nothing really changes in the correlation matrix. You get another flat line. Model 3 and Model 5, we're changing that timing between the spikes. And we see that we could get this slow ramp up in the integrated Betti 0 here for Model 3. Whereas in Model 5, we get this ramp up and then a little bit of a bounce back into a stable plateau when the spike timing is a little bit closer. What's really interesting is also this Model 4, which I think kind of looks like this activation 2, where the neurons are all firing at the same time, but they have different amplitudes, different like amount of calcium or fluorescence that's happening. And there we get this really sharp increase instantaneously. Yeah.
This is just a quick sanity check on the plots. So there's two plots at the top, the data ones there. The reason that you have a ramp up before the onset, is that because the sliding window is sort of leaking into the onset?
No, actually, so the sliding window, sorry, I should have said this. It's a causal sliding window. So I'm indexing time with here I am and what's going on behind me. So our criteria for detecting onset required a certain number of the neurons to be active. And so because they kind of turn on at different times, there is this, slow ramp up of maybe you have an assembly of, let's say, 40 neurons. Some of them are going to go on before we have enough of them to call it an activation. So I think that's what's kind of happening there. Yeah.
Thank you.
Awesome. So so we were able to capture, I would say, 3 of these 4, but what we weren't able to do with this toy model, just changing the amplitudes and the spike timing, is this really rapid rebound back to baseline. A next question is, how prolific are these kind of shapes of these integrated Betti curves across all 5,000 activations? Do we really even care about them? And the answer is, yeah, I think we should care, because when we do k-means clustering, we get out 6 clusters of these integrated Betti time series. We're doing the clustering on these time series, integrated Betti 0 time series. And we see these kind of dynamic profiles reappearing, including this cluster with this rapid increase and rapid decay back to baseline. And we could see how these are organized in like PC space, that they really genuinely are clustered, but they won't necessarily be like separable by like nice hyperplanes, so to speak. And we also see that the principal components are picking up things like the average of the integrated Betti time series, as well as like the Betti 2 is, I think, looking at the rise, and Betti 3 is looking at the fall, or like the difference between the rise and fall. So they're picking up the like basic like Fourier things that you would think that they should. And from there, we wanted to know what's going on with these different types of dynamic profiles, these different activations that are happening in the brain. Why are there these different behaviors? What's going on possibly biologically? And so we looked at a few different properties of the assemblies being activated. One thing we looked at was, what's the size of the assembly? And we didn't really see any trends across these different clusters. They all seem to have roughly the same, spread of sizes. But when we looked at activation length, we saw that these green, blue, and purple clusters have, and even the yellow too, but especially the green, blue, and purple, have much longer on average activations than these top row of these dynamic profiles. And another thing we saw is that the participation ratio, the percentage of the neurons in the assembly that are active, is also much, much higher for these like lower row dynamic profiles. So we wanted to tease into that a little bit more. What's going on? So one thing we looked at was what's happening before onset for 2 seconds before onset versus what's happening for 2 seconds after onset. And if we look at the participation ratio for the 2 seconds before onset, all of the clusters have similar enough participation ratio, but it's really after the onset that there's this split between the bottom row and top row of activation profiles. We don't see any difference in the excitatory activity or excitatory inhibitory ratio. That balance stays the same across the activation onset. But what we do see a really dramatic difference in is the cholinergic activity after onset. Before onset, we see all of the clusters have similar cholinergic activity, but after onset, this bottom row of dynamic profiles has much more cholinergic activity. And so that kind of has led us to this hypothesis that maybe these cholinergic neurons are recruiting other neurons in the activation to participate, leading to higher participation in longer spontaneous activations, which I think is pretty cool, maybe something for attention. So that's the kind of biological story. And then on the philosophical side, we see these different dynamic behaviors that the same assembly can have. One thing I don't know if I mentioned so much was that one assembly has many different activations over the course of the recording. I would say maybe on average 10. The same assembly has activations that can fall into different dynamic profiles. So that assembly can behave in different ways depending on the time that it's active. So what this is suggesting to us is that there is a need for high dimensionality in order to hold all of these low dimensional behaviors, because we don't want them to all overlap on each other. One assembly is not going to just do one thing. It's going to have the option to do multiple things. And for that, we need some space for these different pathways. So that's, in some sense, a secondary need for more dimensions. The first one is that we already have these 2,000 neurons in the tectum. When we're doing this analysis, we're looking at just assemblies, and that's already a dimensionality reduction to just like one subset of neurons. Something that we didn't look at in this is that multiple assemblies could kind of be coactive at the same time or trigger each other's activity. And so this inter-assembly dynamics is also a really interesting question. And so going from the full tectum to an assembly is a dimensionality reduction. From an assembly down to a different dynamic activation profile is another dimensionality reduction. Nonetheless, for these things to all exist in the same space, we're proposing that you need some, higher dimensionality to hold it all. So that's kind of where this story is for now. Like I said, there are these really interesting inter-assembly dynamics, and the data set comes with an experiment, and but we haven't even looked at this. It's just like before the experiment. So that's what I have to say on this project. I'd love to take a pause, take questions, and then with any extra time, if I do have it, just flash a little bit of fresh research.
Questions?
No, oh, yeah.
So thank you for your lecture. So I have two questions. The first one is, does the Betti numbers tell us the spontaneous activities of the neurons? It's like if we have the zero dimensional Betti numbers when it's 2, does it tell us that there are 2 groups of the neurons of the spontaneous activities?
Oh, okay. So I want to say not quite. So I think there are 2 things going on. So I'm going to go way back to the beginning. So here, when the Betti curve plateaus at 2, that is telling us that there are two connected components in our filtration.
Yep.
So that two is a two connected components. When we do the normalizing of the Betti curves, this 6 is going to be, we're going to normalize everything to start at 1. So then the integrated Betti values can never be 2. They're always going to be a decimal because it's going to be less than 1. It's going to be the area under a curve that only decreases and starts at 1 and it ends at 1. So the immediate values coming out of the integrated Betti values aren't going to tell you like 0.2 does not have its own unique meaning, but what 0.2 has a meaning to is like relative to this gold line here. Sorry, it's a little hard to see. This is the integrated Betti 0 value for a random symmetric matrix. So when I see this curve starting close to that value, it's telling me that maybe before onset, the subcluster structure is kind of random, like a random matrix. But as the activation onset approaches, it's becoming less random. So it's less of a quantitative thing and more of a qualitative thing at this point. Not just less random, but it could have become less random by going down too, right? This went less random by going up. So what this is also saying to me is this is less random and it has more plateaus because a higher Betti 0 and more plateaus means more subcluster structure. So I'm not getting an actual number for the number of subclusters or how long they persisted in the filtration, but I'm just getting this sense of more. Yeah.
Thank you. That's very clear. So the second question is, do you have any packages to?
Yeah, I should. Oh man, that's what's missing from this talk. That's the thing that needed to be in here. I don't think I have this anywhere, and that's a shame. Let me write it on the board. Yeah. Yeah, that actually like. I don't know what's actually capitalized or not. And this is gonna do. Okay, so yeah, building off of code that Chad wrote, and I should have shouted him out earlier, but I took this MATLAB code, ClickTop, and I ported it over to Python. It's called PyClickTop 2023. You could find it on my GitHub page. It's NerdNick. And what that code does is it really just has wrappers so that the user can just put in a symmetric matrix and get out Betti curves. Yeah. So yeah, that's there, and it's hopefully super user friendly. And if you have any questions, you know, I'll be here. But yeah, it should be like plug and play, and you could probably get it going. Like, yeah.
Thank you so much.
Can you repeat again how long were the time series and why you chose half a second for the sliding window?
Yeah. So the whole recording was an hour long, and that was 54,000 time points. Each assembly was active for, on average, 500 time points. So out of 50,000, only about 500 of those were when the assembly was active. So when I was doing the correlation matrices during activation, I would take all of the on time points and just kind of use those time bins. For the sliding window, we looked 2 seconds prior to activation to 2 seconds after. We did look further, but the reason we chose 2 seconds around the onset is because the decay rate of the calcium that we're using in the is there's something, a GCamp 6F. It's like a fast one at the time, and it has, the decay rate is about 3 seconds long. So the idea is that the calcium like fluoresces, and you could kind of see that fluorescence for about 3 seconds. So our time series, when we have that activity and then the decay, that decay is like non-trivial for about 3 seconds. So that was kind of where that timing came into play. The half a second window, we actually did play with that, and we showed that this analysis was pretty robust to choice in window size. So I think we looked at bigger windows, smaller windows, and this half a second window, I think it was really just to give us enough fidelity, you know, around like if the window's too big, you can't slide past the onset until you're far away, and then you might not actually be in the activation any longer. Activations were on average like about 3 seconds, some longer, some shorter. I think we have that somewhere here. Yeah, right. So maybe on average they're like 2 seconds, and the long ones are like 4 seconds. So part of the choice of the 2 seconds is coming from just like the activation lengths themselves. Yeah. Does that answer it? Okay. Okay. How am I doing on timing?
I mean, I guess 40s, we're aiming to
going for
stop,
40?
leaving some more time for questions, yeah.
Okay. Okay, so we're gonna like, okay, maybe like we could all just like take a little stretch and like a little breath. Just me, okay. We're gonna totally pivot now. This is just different project, different objectives. We're gonna be looking at circulant structure in network architecture. Oh, I guess I gave 2 title slides. This all happened really fast. Okay, so this is like fresh work. I'm really excited to share it, get feedback. This is looking for specifically circulant structure in neural network architecture. And I think someone earlier was, probably Chad, was like, you know, if you know what you're looking for, great, but often we don't. Here, I know what I'm looking for. I want to find circulant structure. And I'm going to be looking at the Drosophila connectum, in particular the central complex, which is the part of the Drosophila or fruit fly brain that is involved in navigation. So like Chad motivated earlier, lots of circles to possibly be found. But first of all, what's a circulant matrix? My definition of a circulant matrix is going to be a matrix that could be generated by a shift and rotation of a single vector. So you could imagine this one vector here as being the center vector of this matrix. And I'm going to fill out the rest of the matrix just by shifting and rotating up to the top. If like, you know, whatever vector pops out the bottom matrix, that goes up to the top, and then filling out my whole matrix accordingly. And with this, we could kind of think like topologists and think, oh, well, really, that just means that the right and the left are identified, the top and the bottom's identified. And our matrix really now has the structure of a torus. Cute. That torus isn't the way we're going to be thinking about the topology of circular matrices. Instead, what we're going to do is something very similar to the previous pipeline. We're going to take our symmetric matrix as an input to the click topology pipeline. We're going to build our filtration, and we're going to get out Betti curves. But we're going to do that for 2 particular families of circulant matrices. We're going to look at what I decided to call aligned circulant matrices and opposed circulant matrices. And while they look similar, the difference is in my neighborhood structure. So if I'm this neuron on the diagonal here, my nearest neighbors, the things that I'm highly similar to, are near to me in the indexing structure of the matrix. In the opposed circulant structure, my most similar things are the things antipodal to me in the indexing structure of the circular matrix. So 2 different ways of making these symmetric circulant matrices, stick it into the TDA click topology pipeline, build our filtrations. Let's see what we get out. We're going to look at the persistence diagrams, turn those into Betti curves. So in the aligned symmetric circulant case, we see that we start with a filtration that has nearest neighbors connected around a circle. The next step in the filtration is we're just going to kind of fatten up those neighborhoods. And we're just going to kind of start to fill in this circle. So what we're left with in our persistence diagram is one long living one cycle shown here in blue. And in the Betti curves, this is shown as just this one long flat curve that goes to 1 and goes over. I see a little burst of Betti 2 and Betti 3, but really the thing that is saying this is circulant and aligned to us is this one long-lived one cycle. The opposed symmetric circulant case is kind of interesting. After we add the first n edges and connect everybody up to their nearest neighbor, we also get a circle, but the indexing of the neurons on this circle is different than this circle. Sorry, I didn't put that in there. They look the same, but they are different. So for instance, if this is neuron like middle neuron, these are neurons that are like, you know, a quarter of the way across. No, sorry, half the, like all the way across, but like the 2 that are right there. So we have a circle, but it's just a different ordering. And then when we go to the next step in this filtration, instead of creating these triangles that are filled in, we skip 2 neurons and we make this like a hole with 4 edges. And we keep making holes with 4 edges and holes with 4 edges and holes with 4 edges until finally then they start to close up. And so what we see is this burst of a ton of 1 cycles. Then they're going to close up. We're going to have a burst of a ton of 2 cycles, 3 cycles, so on. And we just get like a stupid amount of homology, like the stupidest amount, to the point where we can't even go higher than like 40 on my computer. So this is to say that even if you have something that is symmetric circulant, there are very different ways that they could be. And oftentimes I think we're really thinking about this aligned symmetric circulant case. So that's what I'm going to be focusing on. So we saw that we have this one long-lived Betti 1 as our main signature. This holds across different sizes of matrices. So that's what I'm showing you here. And we can in fact see how like the birth time and the death time and the lifespan changes as a function of matrix size. And not only can we just see this empirically, these are things that like we can write out and prove, just say this is what the filtration is. We know it. So this is kind of what we think about for this aligned circulant matrix structure. And now we want to see if we could see it in data. And so this is a data set that is open source. I'm very excited about this. Yeah, just like about, again, the opportunity to work with really cool data. So let me kind of ground you in this data set and then tell you what we see. We have the Drosophila, that's our fruit fly. Genelia and is Howard Hughes Medical Institute neuroscience research facility. They, along with others, produced the first partial connectome of the adult female fly brain. It's called the hemibrain data set, and they were able to reconstruct from, I believe, electron microscopy, the neuron, like imaging of the neurons and their different synapses to each other for this part of the brain. This part of the brain includes a region called the central complex that I believe Chad and Nico also spoke about earlier. And the central complex is made up of a few main pieces. 2 of the ones that we're gonna care about today are the protocerebral bridge up here and the ellipsoid body, the ring-like shape down here. And making up these different substructures in the central complex are a bunch of different types of neurons. And the different types of neurons we're going to be looking at are the delta 7s, the EPGs, the PENs, and the PEGs. And so what we get from the connectome data set is a matrix of synapse counts. How many synapses as a presynaptic neuron do I have onto a postsynaptic neuron? And so here my rows are my presynaptic neurons, my columns are my postsynaptic neurons. They've been sorted by their cell type. And what I could then do is think about each of these 16 different subnetworks as a presynaptic to postsynaptic subnetwork. On the diagonal, I have recurrent subnetworks because I'm looking at connections within the same population. Outside of the diagonal, I'm looking at feed-forward networks where I have distinct presynaptic and distinct postsynaptic populations. Yeah, none of these are symmetric. They're just giving me synapse counts from, again, rows indexing presynaptic, columns postsynaptic. And then what we're going to do is we're going to look at each of these subnetworks separately. We're going to think about that as just like one pre-post subnetwork. We're then going to take the neural correlations and do our TDA pipeline, get barcodes, and get some scores for circulant structure. But we're going to do this in two ways. From this subnetwork here, the EPGs to the delta 7s, for instance, we're going to compute neural correlations from the rows, looking at the similarity of the rows. Again, we're just going to do a dot product because this is, again, non-negative data, right? It's 0 if there are no synapses from me to a neuron and positive synapse count otherwise. So dot product between the rows is going to give us a correlation matrix between the presynaptic neurons, in this case, EPG neurons. So this correlation matrix here is telling us how similar are EPG neurons based on their output structure to delta 7. Here, I'm going to do the same thing, but with the columns. Dot product between the columns is going to give me a correlation matrix on the postsynaptic neurons, in this case, delta 7. And this is telling me how similar are the delta 7 neurons based on their input from EPG. Cool. So that's how we're getting these two similarity matrices. We know the pipeline, we know barcodes, and we could say, hey, one long-lived bar, one sort of long-lived bar, but it's the only thing there. And we're going to do this for each of the 16 different subnetworks. And for each subnetwork, I'm actually going to do it 10 times. The data is not going to change. The data is the data. But what can happen is, especially in sparser connectomes, you can have zeros in the neural correlations, or sometimes you could have ties in them, but more likely it's ties on zeros. And that can lead to randomness in the TDA statistics. So we want to be able to see when we have randomness from these ties in the correlations versus, versus just adding a little bit of jitter when I repeat this 10 times. So that's what you're going to see on the next plot. So for instance, just zooming in here, these are like 10 runs of the same matrix, and there are probably not ties in that correlation matrix because the only difference is jitter. For this like black or this like purple subnetwork over here, they're spread out, so there are probably a lot more ties in that matrix actually, maybe coming from these zeros. Okay, so what do we get out from this analysis? Well, first thing we wanted to do is we want to take these barcodes and give them like some type of circularity score based on the thing we see from the TDA. So from what we saw with the aligned circulant structure, we want a single long-lived bar. So one thing I'm going to look at is how long is my longest living bar. That's going to be my first statistic. That's on the x-axis. My next statistic is a singularity ratio, which is kind of saying how much longer is my longest bar to my second longest bar. And so that's going to be on the y-axis. And my claim is that, you know, using my own eyeball cutoffs, that things in this upper right corner are things that have a single long living one cycle because it's both singular and it lives for a long time. And what we see here is that this the correlation on the postsynaptic neurons in this EPG to delta 7 subnetwork has like really strong aligned circulant structure, but the presynaptic correlations on the EPG neurons, we saw that bar was not as long living. That's kind of right on the edge of what I'm counting as circulant structure. So I want to zoom in on this a little bit more, and we could see that there are certain subnetworks, each subnetwork has a different color, that do have strong indication of subcluster structure. It's hard to see here, but the two colors are referring to the same subnetwork. And one is just presynaptic, one is post. They're different symbols, but it's all blurred out in the PDF. But just to kind of show you a little example of like what could be happening here in red, where we really don't see indication of circulant structure. This is the delta 7 to delta 7 neurons. We see that there really is no long-lived one cycle here in the barcode, or sorry, the Betti curves. There isn't in the barcode either. And this kind of looks like rank 1, low rank or something. So that's pretty interesting. So there might be a sense that things over here could be low dimensional, low rank, something. Here in the dark gray color, we have the PEG to the EPG. And again, we don't see in the barcodes any indication of singular structure. So I think that's a good indication. What these magenta lines in the barcodes and the Betti curves are showing is the point in the filtration after which edges are added between neurons with zero correlation. And that was important because there were times where I was like, oh, maybe down here we have things that we have one cycles that live for a long time, but the singularity score is low. Maybe we have multiple one cycles. Maybe there are like 3 of them there, and that's what I'm seeing. How cool would that be? And sorry, I'm going to zoom in on this. It's like, yeah, there, look at 2 bars, 3 bars, but all of this is happening on the zeros and the correlation. So this is just structure and the randomness. So, you know, important to look at your data, right? You know, like look at it every step of the way. But just another example of it doing a really good job in this case, you know, don't go off and like, you know, I shouldn't be too bold and say, this is what's happening here and this is happening here. But here I feel confident because when we look at this, we really do see these really long single bars. So that's pretty cool to me. And I think this figure kind of summarizes where my findings are so far. So I'm just going to walk through it really quickly, flash you the next one and wrap it up. Okay, so because I didn't give you a color key, these are the networks, the subnetworks that I've identified as having aligned circulant subcluster structure. The arrows go from presynaptic to postsynaptic population. What I did was I did some normalizations and controls, shuffling the values, normalizing, binarizing the connectome. And that allowed me to say that the arrows that are fat are sensitive to the specific synapse count, where the arrows that are thin, they're robust to like binarizing the connectome, and I still see that circulant structure. So that's pretty wild and interesting. The dashes are showing me when there's a discrepancy, like with this EPG to delta 7 network, a discrepancy between the pre and post neural correlations in terms of their circulant structure, which is pretty interesting because that wouldn't arise in a Dowker setting, but we're not doing Dowker. I think that's really interesting. And I guess I just want to like put up there kind of these bigger questions that I now have, which is what's leading to this discrepancy in the TDA between the pre and postsynaptic correlations? Skipping down to question number 3, how does this RIPS filtration, this order filtration I'm doing relate to a Dowker filtration directly on the synapses? And middle question number 2, which I didn't show you anything, but it's supremely weird. But maybe it's not weird, it's just interesting. Right now I'm taking like a single presynaptic population and a single postsynaptic population. If I were to combine subpopulations and say what's happening from EPG and delta 7 to EPG and delta 7, I lose the TDA signature of circulant structure. And I want to understand that better. So thank you for the abundance of time. That's it.